Well, I wanted to thank the organizers for inviting me to this fantastic conference. All right. So, today I'll be talking to you guys about a model that we've developed in the lab in the last few years, which is intestinal organoid. And this body of work, we've used that in order to look at the potential direct effect of hyperglycemia on gut barrier function. And I'll start by acknowledging, first of all, I need to acknowledge Kiki Crawford. So he recently graduated, he was my PhD student, and all the work that I'll be sharing here really was spearheaded by him. And you can see that this picture was taken at the beginning of the summer. He's holding his first newborn, and he just recently got a teaching position at the University of Southern California, so lots of good, exciting, and new things on his future. And obviously the rest of my team, and then just funding for this project, which was the Center for Companion Animal Health at UC Davis and the Every Cat Health Foundation. So, as I probably don't need to tell you guys about diabetes, how common it is, both obviously in the human population, but also in the cats, right? So it's one of the most common endocrinopathies that we see in our cat patients. And obviously, diabetes is a very complex, multifactorial disease, but at the bottom line, what we see is persistent hyperglycemia. Regardless of the initial etiology, this is what we're dealing with, persistent hyperglycemia. And since it's such a chronic disease, and because glucose homeostasis is so important to many organ systems in the body, we see lots of comorbidities associated with chronic persistent hyperglycemia, one of them being diabetic enteropathy. And again, this is a complex manifestation of the disease, and we have factors such as oxidative stress, neuroinflammation, reduced nerve growth factors, vasculopathy. All of those are known to contribute to that syndrome of diabetic enteropathy. But surprisingly enough, there is not a lot of knowledge about the direct impact of glucose on epithelial permeability, because it's really hard to parse out the direct effect of hyperglycemia in vivo models. And for in vitro models, we don't have great tools for that. We have intestinal cell lines such as KCO2s and other cells which are human, and they're derived from cancer cell lines. And then what people have done in the past is also treating with glucose, but that would be on the apical side and not on the basolateral side, which would recapitulate hyperglycemia. The other thing that we were interested in, we had some interest in fenofibrate. So fenofibrate is a PPAR-alpha agonist, and PPAR-alpha is a transcription factor. And it's a drug. It's a drug that's been on the market for a long time. It's FDA approved. But more recently, we've seen, so through work of my collaborators, we saw that fenofibrate, so their model, they had a model of induced diabetes in dogs, and they treated these dogs with fenofibrate, and they saw significant improvement of gut structure and permeability in that in vivo model. And also some other work that I was involved with collaborators in the SIV model, we saw that also PPAR-alpha and other so fenofibrate and other PPAR-alpha agonists really improved barrier function in that very different but still model for increased gut permeability. So we wanted to test the hypothesis whether hyperglycemia directly induces intestinal barrier dysfunction in diabetic cats by disrupting the intestinal epithelial tight junctions, and further, whether that could be reduced by fenofibrate. In order to do that, we've generated feline intestinal organoids, and the way that we do it, we used adult stem cells. So we got biopsies of small intestinal biopsies from cats, and we've used published protocol to generate those organoids you can see the way that they look here. So they're more spherical. So I can tell you, being working in the past 4 years with organoids. Organoids from different species have very different morphology. Some of them would be more spherical. Some would have that more typical kind of lobulation, where you can actually physically see kind of villi and crypt domain. But for the cats, we got those really spherical organoids. So what we did is that we treated them very consistently 4 days after we passaged them. We treated them with glucose, and then we had some controls, right? Because we wanted to make sure that, for example, that whatever effect we're seeing is not due to the increased osmotic pressure generated by this hyperglycemia. So we used mannitol as a inert sugar. We wanted to make sure that it's because of intracellular movement of glucose into the cell, so we blocked it by a GLU2 inhibitor. And then we had our treatment group, right, which was glucose plus fenofibrate. Our readouts were also pretty simple. So the first thing that we looked at was permeability assay. And if you want to look at the details of that, we recently published a cow intestinal organoid paper where you can have all the, the details of the way that we look at that. But we looked at the permeability with a FTSE-dextran assay. We then looked at tight junction morphology, looking specifically at ZO1. And then we did some RT-PCR, targeted PCR, and also we looked at cell turnover. So I'll start by permeability, because that was really the main question that we want to ask, whether hyperglycemia directly, regardless of inflammation, which is part of it, regardless of oxidative injury, whether hyperglycemia directly has an effect on gut barrier function in cats. And the answer is yes, right? So you can see here, so this is a normal organoid. So we bathe it in FTSE-dextran, and then we wash it, and then we image it with a microscope, with a fluorescent microscope. And what we do is that we measure the fluorescent intensity around the organoid and then within the organoid, and we derive the ratio. And in each culture condition, we have dozens of those organoids. So each data point that you see here is an average of an experiment. So we've done that a lot of times. So Kiki spent lots of time measuring the fluorescent intensity around all of those organoids. So this is a normal organoid. Outside we have residual FTSE, but inside you can see that it's pretty pitch black. So in a normal organoid, the dextran can't trickle through the tight junctions. They're very tight. Whereas with glucose, you can see that we have lots of green kind of inside, and that's reflected again. Look at that, that's much higher, and obviously statistically significant. And just so I don't have an image of mannitol, but the mannitol was as dark almost as the untreated. And here you can see what happened when we used glucose but blocked its entrance into the intestinal epithelial cells with a GLU2 inhibitor, pitch black inside. And exciting enough, we saw that when we treated that with fenofibrate, again, it was insignificantly higher than baseline. So, maybe there was a slight trend, but it was statistically insignificant, and it's definitely much lower and statistically significantly lower than the glucose. So that was very exciting to us. So now that we've seen that, we try to figure out what's really happening here, right? So the first thing that we did, because we kind of developed that method and it makes sense to look at tight junctions, we looked at tight junctions. And the way that we do that is by staining for, again, ZO1, which is a major tight junction protein, and we look at the intensity of it and also the structure of it, right? So this is here, right? This is baseline, and you can see that those lines are pretty bright and that they're pretty straight. And we can measure the straightness or tortuosity of those lines. And if you compare the untreated to the glucose, you can see how tortuous those lines are, right? And when we quantify it, we see that it's significantly higher. So those tight junctions, their morphology, their structure gets completely messed up, right? Instead of being nice, straight rows, it's becoming all tortuous. And when we use the inhibitor, when we use the fenofibrate, that all goes away, so it looks very much like control. So again, very excited about that because we know that tight junction play a significant role in that. So to remind you, we know that PPAR PPAR alpha is a transcription factor. So we thought that fenofibrate does that by regulating gene transcription of those tight junctions. When we did the PCR, everything was exactly the same. So we didn't see any alteration in gene transcription, which was kind of a head scratcher for us, like what's going on here? So if fenofibrate does not alter gene transcription, what does it do? So we know from previous work, we know that fenofibrate reduces PKC alpha activation, PKC protein kinase C activation in platelets. And as a reminder, platelets, they don't have nuclei, right? So, so fenofibrate does something in platelets without altering gene transcription. We also know that increased PKC alpha activity is associated with increased intestinal permeability. And that increased concentration of DAG, which is a PKC activator, is a byproduct of glucose metabolism, right? So all of that kind of made really Kiki hypothesize that hyperglycemia activates PKC alpha function, which is inhibited by fenofibrate, so in a non-gene transcription modulation way. So in order to do that, he did some Western blots, and in a nutshell, that's exactly what he found, right? So he found that when you look just at PKC, the protein itself, the kinase itself, there is no difference between our various treatment groups, as you might expect. But when we looked at the phosphorylated proteins, we saw a big increase with glucose, and that was truly inhibited by fenofibrate. So if to summarize, this is what we are currently thinking about this system. We think that hyperglycemia, right, and just, in our model, I didn't emphasize that, but we expose our organoids to glucose through the basolateral surfaces, right? So those spherical structures, their lumen is in the inside. So the high glucose concentration is really mimicking the hyperglycemia that intestinal cells see in vivo. So we think that we have hyperglycemia that causes glucose to leak in through the GLUT2 transporter back into intestinal epithelial cells. Increased concentration of glucose are leading to increased concentrations of DAG, which activate PKC alpha. Once that happens, we don't know how, but that messes up the tight junctions. And that allows for things to flow through those tight junctions in both directions. And that's something that we don't want to have, right? We don't want water and good stuff to go into the lumen, and we definitely don't want bacterial products and stuff like that going into the body, right? And the exciting part is that it seems like fenofibrate is working right here at the PKC alpha. So fenofibrate, while it's a PPAR alpha agonist, it blocks directly PKC alpha and does not allow that to do those negative effects that we've seen. So this is just what I've kind of talked about, our proposed model. And then I just want to talk briefly, about the organoids of an in vitro model system. And the big advantages of intestinal organoids is that, as veterinarians, we want to work with the species that we care about. So we can, we don't need to work with human cell lines or mouse cell lines. We can actually work with a species that we're interested in. In organoids, we have all the, for people that don't work with epithelial cells, with intestinal epithelial cells, we think that they're all the same, but they're really not, right? We have the enterocytes, you have the goblet cells, you have the stem cells at the bottom of the crypts. There is lots of heterogeneity of cell types within the intestine that are not represented in other in vitro systems. And we have more control over apical versus basolateral. So these are all great advantages of intestinal organoids. The negative is that I think that our culture system is definitely not still well optimized. So I'll share with you that we have tried to generate, different species. They work differently. Our cows, they're easy to work. They're great. Cats are not as easy, right? So we, some of our attempts to generate organoids really weren't successful, and with that cell line, we just got lucky. I feel like that was, I don't remember, I think it's like 1 out of 4 attempts. So our culture conditions and the way that we need to, the protocol for generating those organoids, they're definitely not optimized yet. So there is definitely work to be done there. So that would be all, and I'd be happy to take any questions.
Any questions? Okay, any hard questions?
What other applications, so for instance, in animals with severe colitis or enteritis, some pathogens, including bacteria, can go through those tight junctions. Do you think this the agent that you explored could be used to prevent or like administer while animal is sick to?
So again, so we've seen that not only in diabetic models, but we also have seen that, through my work with the SIV model, there is increasing evidence that that's really helpful there. But I don't know what's the mechanism there, right? But it seems that it's definitely helping in two very different models. And sure, whatever, when you have an issue with barrier function and leakage, yeah, then that definitely could be an issue. So the other thing, taking that and translating that to, diabetic patients, obviously the goal would be to cure diabetes, right, and, nip it at the bud. But other thing that we need to find is probably something that does not get absorbed systemically, something so a PPAR alpha or, fenofibrate-like substance that will act upon those intestinal epithelial cells and would not be absorbed systemically and have, potential effects that we're not interested in. Yes.
Amir, great talk. In my prior life, I ran a preclinical animal model business for a lot of human drug discovery, so this is near and dear. But one of the big arguments against the preclinical using in vitro models is there's no intact immune system to interact. Have you thought about how you could address that problem?
Absolutely. It really depends, I think, what kind of model are you interested in, right? So, there is different levels of complexity, right, that would fit, the question that you want to ask. I know that there are people that are trying to incorporate immune cells into those organoids. So, for example, if you make those organoids from iPSCs and not from adult stem cells, you'd actually get the stromal component as well, which we don't have. And then people are trying to add in hematopoietic stem and progenitor cells in order to get those immune cells for, for models such as IBD, et cetera. But we actually, for us, that was an advantage because we wanted to, to parse out, to isolate the effect of glucose on epithelial cells regardless of inflammation, because we know that inflammation also disrupts the gut barrier.
All right, thank you so much. Enjoy the rest of your day.